So previous lectures have examined evolution at the genetic level using relatively simple examples, oftentimes assuming that a trait is associated with a single gene locus. But then I started ramping up the examples, talking about how you can have interactive evolution among a couple of gene loci and the effect of linkage in those situations. And that led to discussions of why sexual reproduction is so important and recombination is so important. Now we're going to ramp it up again today. We're going to talk about the fact that many traits, if not most, are what we call polygenic traits. Traits that the phenotype is based upon the expression of many gene loci. Let me first preface this video by stating that quantitative genetics is a really expansive and complex topic, so this video is really going to be a relatively superficial summary of the field. So throughout the semester I've been giving you examples of traits associated with genetic basis, so heritability, but I've really kind of focused on relatively simple single locus traits. We did start ramping it up by talking about traits that might be influenced by two loci when we talked about linkage disequilibrium. But most traits really are controlled by the interaction of many more genetic loci. And there's also an environmental effect associated with the expression of various phenotypes. So the study of quantitative genetics is the study of these polygenic traits. One characteristic of polygenic traits is they show continuous variability with regard to phenotypic distribution of a trait. So we don't have a set phenotypic category like when I was giving you some examples of some relatively simple traits in the past. Like think about the typical Mendelian traits of flower color. It's either purple or white, or tall or short. And when we talk about polygenic traits, we don't have those specific categories. So let's give you an example and show you why the more gene loci you have, the more you're going to approach a normal curve or a bell-shaped curve with regard to the phenotypic variation in a trait. Let's say that you have just one locus, a real simple example, and we're going to use the example here giving incomplete dominance. So you mate a short individual and a tall individual. So it looks like they're pretty set categories here, but the homozygotes in both cases, when you breed them, mate them, and you produce heterozygotes, you see that you get a third category. You get this intermediate, and then the heterozygotes, when you crossbreed those, you end up sorting them into the three basic categories because of incomplete dominance. So we do have just three categories here, but we're just talking about a one locus trait. Now let's talk about if we have two loci that are following incomplete dominance. Again, you may start off with the two categories, short and tall. The first generation, when you breed these, is going to be intermediate, so we're just going to get the intermediate height, the middle sized. But then when you breed the heterozygotes, you're getting more combinations. So now you have kind of like the intermediates between the medium and the short, and then you have the intermediates between the medium and the tall. And then if we bump that up to 6 loci, wow, now clearly we don't have set categories. We clearly have a bell-shaped curve with continuous distribution associated with the height of these plants. So the point is, the greater the number of gene loci that are involved in the expression of a trait, the smoother that phenotypic distribution is going to be. So it sorts itself out into a bell-shaped curve. Here are a couple of examples of this. So human height is a classic example of a polygenic trait. And here we have the University of Connecticut marching band lined up, both males and females. So the males are in the blue and the females in the white. But together, that's a pretty good bell-shaped curve there. And even within both sexes, there is a bell-shaped curve if you line them up by height. Same thing with swimming speed. The study looking at swimming speed. There's a difference between adult men and women, but within each there is a nice bell-shaped curve. So there isn't just like two swimming speeds, fast, slow. And again, this bell-shaped distribution is due to the distribution of alleles for the multiple gene loci that are associated with the expression of this trait. So think about it. If you just think about it with regard to human height. We don't even know how many gene loci are involved in height, but let's just say that there are 50. It's very unlikely that in each of these 50 loci, there's maybe just 2 alleles, one that contributes to the degree of shortness and tallness. It's very unlikely that you're going to get all of the tall alleles at every single gene locus. It's also unlikely that any individual is going to get all of the short alleles for each gene locus. So most people are going to have some various combination of tall alleles and short alleles to give them an intermediate height. Some individuals, if they have taller parents that had more tall genes, well, they're going to get a greater proportion of tall genes. Another thing that can contribute to a more continuous distribution among traits in a polygenic trait is the effect of the environment. Phenotypes of polygenic traits are also influenced to some degree by environmental conditions. Now, this has to do in large part with the degree of heritability associated with the trait, and that's actually going to be one of the challenges in any kind of quantitative genetics study. But here's an example of a plant, goldenrod, in which individuals with the exact same genotype, so they're clones of each other, they don't have the exact same phenotype when they're grown in different environments. And the greater the environmental difference, the greater the potential phenotypic effect here. At lower elevation, you get really tall plants. Same genetics, as you go up the mountain, you get smaller and smaller plants. So this is an example of what we call phenotypic plasticity. And we'll be covering this in future lectures. Okay, so let's look at the potential for polygenic traits to evolve and how we can actually study this. Now, we're going to cover in this lecture 2 broad kind of approaches to quantitative genetics. And I'm going to start with some more modern techniques, looking specifically at the gene loci that are associated with phenotype and phenotypic variation. So if you look at the Mimulus phylogeny, these are flowers that have different coloration and shapes. And 2 of these species are fairly close related, Mimulus cardinalis and Mimulus lewisi. They differ in a number of traits in both coloration and flower shape. The cardinalis flowers are red and tubular, and this is a common pattern that we see in flowers that are co-evolved with hummingbirds to attract hummingbirds as pollinators. Lewisi, on the other hand, is purple, relatively flat, and it's got these yellow spots associated with the landing pad. This is a common pattern that we see in flowers that are adapted for bee pollinators. And if you compare the phylogeny of this entire group, it appears that this kind of flat and yellowish flower is the plesiomorphic condition. Plesiomorphic means the primitive condition. So either kind of a yellow or a white flower. And that red flowers have evolved 2 times. So you can see one is associated with cardinalis up here, and then we can also see the evolution of the red tubular flowers evolving down here. So there's been convergent evolution of traits to produce more tubular red flowers for the attraction of hummingbirds. And this is a good example of using a comparative method. We're going to cover this in future lectures, but this is a good way of using phylogenies to reconstruct the potential pattern of evolution of traits. Well, so this is basically just showing you what the phenotypic evolution is associated with flower color and shape. Now the question is, what are the QTL responsible for these differences? QTL stand for quantitative trait loci. So what's the genetic differences between cardinalis and lewisi that produce such different flowers from a common ancestor that had the same genes? Well, luckily these 2 species of flowers are close enough that there hasn't been reproductive isolation. You can still interbreed them artificially and produce viable offspring. So if you cross individuals from both of these species, the original species, a cardinalis on the right, lewisi on the left, the F1 generation is a really good intermediate. So it's starting to get more tubular. It's closer to red than the light pink, pinkish white. The kind of landing pad coloration is changing as well. And all of the flowers in the F1 generation look pretty much like this. Now, if you take individuals from the F1 generation and you crossbreed those, then look at what the variation you get in the F2 generation. I mean, you get everything, all these intermediates in coloration and shape, with, interestingly, some rarely that look just like Loci or some that look almost identical to Cardinalis. So what's going on here? Well, what's happening is that at each locus associated with the production of some aspect of these flower shape and color, each of these species are homozygous for just one allele. So they're kind of fixed in the population for this one allele within each species, such that the hybrids in the F1 generation are getting one each, one allele from one species and one allele from the other to produce this nice intermediate. But at all of these different loci then, when you interbreed the F1s, wow, you're getting all combinations of the Loci alleles and the Cardinalis alleles at the different loci. And so you're producing all of these variable types, including some that just happen to get all the Loci alleles and some that get primarily the Cardinalis alleles. So in a study like this, one of the goals is to try to figure out how many gene loci are responsible for this trait variation and where might they exist in the genome. We can use marker loci that have known locations on specific chromosomes and the pattern of linkage disequilibrium as a tool to help us uncover these patterns. Now, the marker loci are neutral loci, genetic regions that are not related to the trait that we're studying, but we happen to know where they are. So we just know what chromosome they are and what position of the chromosome a number of these markers are. And if we get a pattern like this, where we see the Loci alleles being linked to the Q stands for the quantitative trait loci associated with Loci versus Cardinalis. So the yellow one here is Cardinalis, the black one here is Loci. If it happens to be closely linked to a very specific allele at Loci in this species and Cardinalis in this species, then in the F1 generation, this is the only combinations you're going to get. Okay, so you're going to get these intermediates, right? We're going to get, here we have both allele associated with a gene locus at the flower determining locus, and then closely linked to it, we get the exact same locus associated with one of the marker alleles at Cardinalis. Same thing we're getting with Loci next to it. And then when you cross the F1s to get the F2s, these are the only combinations you're going to get if, importantly, there's very tight linkage between the marker locus and the trait production locus. Such that if you look at the phenotypic distribution of the marker loci that are homozygous Loci, they're going to have a characteristic, and I'm going to show you on the x-axis down here in a minute. So this is basically showing you flower that looks like Loci. This is a flower that looks like Cardinalis. This is a flower that looks like an intermediate of those because it has the marker locus L and a marker locus C. So it's heterozygous at that marker locus. So we know what the marker loci are, and if it fits nicely into these distributions linked to a specific phenotypic trait, then we know that somewhere near that marker locus, somewhere linked closely to it, there is a locus that is responsible for the production of this phenotype. So it's helping us identify it, like, this marker locus isn't doing it, but near this marker locus, there is a gene that is giving us this trait type. Well, what's the other possibility that could be happening here? Let's say that we start off with the same thing. So we have a marker locus and then we have the quantitative locus associated with each species. You see that the production of the F1s, you're going to get the same thing because you're breeding homozygotes of both species. But look at what happens in the F2 generation. Wow, the F2 generation, there's absolutely no association of a specific phenotype between the Loci versus cardinalis or an intermediate and the individual marker loci. There's just no relationship there. So this tells us that that marker locus didn't give us any information. It doesn't tell us that there is a gene locus near that marker locus that is associated with the production of this trait. I mean, I guess it tells us that there's not one in that area, so it doesn't tell us where one is, but it might tell us where one isn't. We're using a QTL mapping. There have been QTLs discovered for 9 flower traits, and it explains about 25% of the phenotypic variation across these species. One of them, one specific trait locus, quantitative trait locus, is associated with a change in flower color, making it either purple or orange. And that's what's being shown down here. So here we have the wild type Loci. Here we have the Loci with one of the cardinalis genes at that locus. Just that change, at that one gene locus, made this change. Now clearly there are lots of others that are responsible for the differences between the flower shape and color between the two species, but this is just one in showing you how this complex trait could change gradually through time, because this isn't going to be nearly as successful as a red tubular flower, but studies did show that this one on the right had an increased ability to attract hummingbirds to it for pollination compared to the one on the left. So it's a gradual step in the evolution of a more adaptive flower for hummingbird pollination. So what QTL mapping does is it provides us an approximate location of where QTL might exist. So we know it's somewhere linked to the marker locus. But then what you have to do is you have to go in and look at the coding loci in that area and look at the protein products that they produce and determine the function of those protein products. This can allow you to establish which specific loci really do affect the trait of interest, and how does protein variation at that locus affect the trait. This type of approach has allowed us to understand a little bit about Crohn's disease. It's not, again, it's a polygenic trait, so it's not a complete explanation for why some people have it and some don't, but you're more prone to Crohn's disease if you have a mutation where you're homozygous for that mutant. So if your parents are carriers for the NAD2 mutant, this mutant right here, they don't have one of the wild types. Well, they only have one of the wild types, but you don't happen to get it. You happen to be homozygous for the mutant, this recessive mutant allele. Again, this is an example of a deleterious recessive trait. Then you're more prone to having Crohn's disease. So that's a brief introduction and summary of what QTL mapping does. Now let's talk about a kind of an older way of studying quantitative traits. In this case, we're looking at polygenic traits, but we don't really know the location of the QTLs or the number of QTLs. In this case, researchers are trying to examine patterns of changes in phenotypes from generation to generation, not really focused on the specific alleles or allelic combinations, but trying to get a correlation between phenotypic variation and the likelihood of traits being heritable to a greater or lesser degree, without knowing exactly what alleles or gene loci are involved. The first step in this is to determine the relative effect of genetic components versus environmental components to the trait variation. Because remember, if a trait is not heritable, if the variation you see, the phenotypic variation, is entirely dependent on variable environment, then selection can't operate on that. And so that has important consequences for evolution. And trying to determine this in quantitative genetics is not easy. So let me give you a simple example of this. Let's say that we have siblings, birds, that were raised in the same nest, and they have really similar sized bills. compared to other birds in the population. The similarity that they share in their bill sizes is closer than they are to any other birds in the population or typical birds in the population. And you may be saying, well, that's clear evidence that they've got the same genes from mom and dad to produce that specific bill size. So the similarity is really due to the same alleles at whatever QTLs are responsible for the production of bills. Well, there's another possibility though. Maybe genetically there's really not much heritability associated with this trait, and that the similarity is due to the fact that they share the same parents. They share the same environmental conditions. They were raised in the same way. Maybe they have big bills because they had really good parents that were very good foragers and brought a regular supply of high protein, highly nutritious food, and that's what allowed them to produce these large bills. And because they got the same food and the same parents, they're very similar. So the challenge is, how do we separate the genetic aspect of determining what your bill size is versus the environmental effect? And in this case, it's really difficult to do. I'm going to give you an example here in a bit how studying birds you could test this. All right, let's break this down to formulas. We're going to talk about measuring heritability in two different ways now. We're going to talk about broad sense heritability and then narrow. The goal is to look at the phenotypic variation we have among individuals, which is VP, and try to understand how much this phenotypic variation is due to genetic variation and environmental variation. So what is the relative effect of the trait that you get in an organism? Is it primarily based upon the different genes that they have, or how they were raised, the different environmental aspects that they had during development? What's the variation of the phenotype, and how much of that is controlled by genetics, and how much of that is controlled by environmental influences? Well, one approach is to try to estimate what is called broad sense heritability. This is the fraction of the total phenotypic variation that is due to some genetic determination. So this is the formula for broad sense heritability. What fraction of the phenotypic variation is due to genetic variation? And this heritability ranges from 0 to 1. So a heritability of 0 means it's all environmental influenced. Heritability of 1 indicates that there's really high heritability, very strong genetic basis to this, and environmental differences really are not going to make much of a difference in the phenotypic trait produced. So here is actually the possibilities. If we looked at, say, height in humans between the parents, which the mid-parent is basically the average parent size, mom and dad, versus the mid-offspring size of the siblings produced. If we found a pattern like this, there's really no pattern there, right? I mean, they've got a line drawn here, but that's just random. That heritability is pretty much 0. Here, we're starting to get a pattern, and we see some slope associated with this. So there's a better chance that tall parents produce tall offspring. There's still a lot of variation around here. So this indicates that there's a mix of environmental influences, so perhaps diet and heritability, that controls the final height of the offspring. But look down here. Boy, we have a really tight correlation. A 1-to-1, we have a slope of 1 associated with tall parents producing tall offspring, short parents producing short offspring. So what's really the reality? Well, if you collect real data, this is what you get. You get a slope of 0.84. So how tall you are is primarily determined by genetics. Is there an environmental influence? Yeah, somewhat, but it's primarily being driven by what genes you have, what alleles you have at several quantitative trait loci. And again, how many, we don't know. We went through an example earlier in this semester where we used this same approach to estimate heritability in the context of, can this trait evolve by natural selection? And we saw that there was a really tight heritability associated with beak depth, kind of how tall the bill is. So parents with really tall bills produce offspring with tall bills, and short bills are parents produce short-billed offspring. Okay, so that's basically just trying to separate the genetic component versus the environmental component. There's a more specific approach in quantitative genetics where we're trying to estimate heritability, not broad sense heritability, but narrow sense heritability. This is a more specific heritability measure, and it is abbreviated as little h squared. So what we're going to do is we're going to take the Vg in the previous equation and break it up into two genetic components, an additive genetic component, Va, and a non-additive genetic component, which is also called the dominance component, and that's why it's called Vd. Now, unfortunately, the naming of some of these leads to a bit of confusion, so let me try to walk you through that and explain the differences between the additive genetic component and the dominant genetic component. But this is basically what we're doing. We're saying our measure of genetic variation in the broad sense, just total genetic variation, now we're trying to be more specific. How much of this genetic variation is due to an additive effect versus a dominant component? And really, h squared is only focused on one of these aspects of genetic variation. We're really only interested in how much of the phenotypic variation is due to the additive genetic component. And the reason for this is I'm going to explain. The additive genetic component is guaranteed to influence the offspring's trait in one degree or the other, make it bigger or smaller, lighter or darker. It is something that that is predictable. So we're trying to figure out what is the predictable genetic contribution to the variation we see in the phenotype. So as I mentioned, the additive genetic component is the part that will influence the offspring's trait. Now, your textbook has a kind of a mathematical explanation for this, box 9.2, for a distinction between the additive and genetic components. Check that out if it helps you understand it better. But I'm going to give you just kind of a verbal description of the difference between the additive and the dominant component, and that's all I expect on the exam. So, as I mentioned, the additive component is the proportion of the genetic variation that will increase or decrease a trait's value in the offspring, independent of mate choice. And that's the key. The dominant component is also a genetic contribution, but whether it's expressed depends on the alleles that you get, that you combine from the individual you mate with. All right, so let's try to understand the difference between them a little bit better. The dominant component is associated with the presence of recessive alleles. So think about it. Recessive allele is trying to say produce this product or don't produce this product if that's what the allele is associated with. But you're only going to get the expression of that phenotypic request if it's combined with another recessive allele, right? So that depends on who you mate with. And you can't predict how often this is going to happen in a population and how that's going to affect the expression of a polygenic trait. So this is called the dominance component, but it's because of a dominant effect, but really it's kind of linked to how often you have recessive alleles that you can't predict that they are going to contribute to a trait's phenotype one way or the other. The additive component, we're talking about the situation there where it's dominant alleles or incomplete dominance, where when you get this allele, you're going to get this degree of increase or decrease in the trait. Now, the reason it's important to calculate this, if you're studying the potential genetic effect of on a trait's phenotype, is the proportion of variation in each component really tells you how well this trait is going to respond to some selection pressure. If the variation is phenotypic variation is due simply to environmental variation, selection can't do anything with that, right? It just depends on the environment that you're raised in, the diet that you have, whether it was a wet condition or dry condition. If, however, most of the phenotypic variation that we see is linked to variation in additive genetic component, then natural selection can really do something with that. All right, so let's come back to that estimate of heritability and how we can do cross-fostering studies to disentangle the amount of variation in a phenotype due to environment versus the additive genetic component. So we do our typical mid-parent study where we look at the mid-parent's value and look at it to the mid-offspring's value, and you say, okay, wow, that's really good. Look at that. we have a really high heritability here, 0.98. And you may be thinking, well, but again, we still have the same problem. Is this because it's the parents, or the parents, or all of these siblings had the same parents in the same environment? So what you can do is you can take half of the offspring from a nest and you swap them with another parent. So you cross-foster them and see if it's the same pattern. Because if you see the same pattern here, it's not what's being shown here, but if you did see the same pattern as what you have up here, that's telling you it all has to do with the quality of the parents, how much food that they're bringing, or the temperature associated with the nest in which they were developing. But instead, what you see is there's really no relationship at all between what mid-parent you got and the size of their bill and the size that you end up producing. So this tells you that there really is not much of an environmental impact here on bill size. This is really a highly heritable trait, and it is due almost entirely to the additive genetic component. So the slope here indicates that narrow sense heritability is 0.98. Now in humans, we can get a sense of this by looking at twin studies. If heritability is high, monozygotic twins should be more similar than dizygotic twins. So if both sets of twins are raised in the same environment, we're controlling for environmental variation. They're raised in the same environment, the monozygotic twins or the dizygotic twins. But if the monozygotic twins are very similar to each other, that tells you that it doesn't really tell you by itself that there's an environmental versus genetic component. But if you look at dizygotic twins raised in the same environment and there's a difference, that's telling you that there's heritability associated with that. They're different because they got different genes. This is kind of a graph. I can show you that down here. So high heritability shows you that the monozygotic twins are really identical. There's more variation here in the individuals that are dizygotic, despite being raised in the same environment. They've got different genes, and so they're different. Low heritability, here we have a situation where there's no difference between monozygotic and dizygotic twins, and if they show the same degree of genetic difference, same degree of phenotypic differences, it can't be explained by differences in the percentage of genes that they have. So let's look at the consequence of having a trait that's got high heritability versus low heritability. We can test the response to some artificial selection pressure by doing what's called a truncation selection experiment, where we try to shift a population that starts off with a certain distribution to one extreme phenotype or the other. So this is a form of artificial directional selection called truncation selection. So let's say that we have a group of organisms where we have a variation in body size and we want to produce bigger individuals. Well, we should truncate this by only allowing this group of individuals to breed. If there's high heritability, the next generation should shift to the right, because we are selecting for larger individuals, and this is only going to be possible if there's high heritability, so that the individuals that are bigger, well, they have many of the big-bodied alleles compared to other individuals in that population. So how fast we'll be able to shift this distribution to the right is what we call the response to selection. So this is the difference between the parental population and the offspring population that we are able to form after this truncation selection experiment. Now there are two factors that are going to determine what R is. So how fast, what is our response to selection? One is how picky we are, S. This is the selection differential, kind of how far we truncate that breeding population. The other aspect of that is narrow sense heritability. Is the difference between the big individuals and small individuals based on environmental component, or is it due to additive genetics that they have? So this is going to be a very important graph for you to understand each of these components and the consequence of their variation. So let's say we start off with a population that's got some pretty normal distribution of traits, and we take this proportion of the population to breed to make the next generation. Well, the entire population we started with, this was their mean for whatever trait we're talking about. Maybe we're talking about milk yield. We're trying to increase the amount of milk that we can produce in some dairy cows. Well, we're only going to let the individuals that produce a lot of milk or the brothers associated with them, and we're going to crossbreed them so there's no inbreeding. But we're going to take those lineages that are linked potentially to greater milk production and have the potential for genes for that. We're just going to let those breed. And the mean of that population versus the mean of that population is our selection differential, the S. So it's how picky we are. So if we only use this many, like if we move way over here, S would be bigger, right? We would have moved it over. If we just let them all breed at random, what would S be? Nothing, right? I mean, you're not being picky at all. Okay, so S is the selection differential is how picky you are in establishment of who is going to breed to establish the next generation. The pickier you are, the bigger that number, and the greater potential response to R. The bigger R will be is if S is large, right? The larger S, the larger potential change in R. But S isn't the only thing that contributes to R. Okay, so here's basically what we're trying to do. We're trying to get this to shift off to the right. If this is the old XP, which is over here, and we're shifting it to the right, this is the new one, we're trying to get R. And the bigger the S, the greater the potential of R. But again, we also have to contribute, we have to ask, what is the narrow sense heritability associated with this trait? All right, so what do I mean by that? What is the potential impact of heritability? Well, think about it. If the heritability is 0, if there's no real genetics, but determining how much milk you can produce, maybe just what diet you have or how the conditions are as far as how hot you get, how much water you get. I mean, if it's all environmental, this is 0. It doesn't matter what S is. S could be way over here. It doesn't matter. You're not going to get any response to selection. The population isn't going to shift. You're not going to get an R. However, if this is a highly heritable trait, that this is close to 1, such that the phenotypic variation between individuals in this original population is due to the fact that they've got really a lot of different alleles at a number of different loci that contribute to the milk yield. In that case, the more picky you are in your selection differential, that's going to be multiplied by something close to 1. And so you're going to get a bigger response to selection. So it's really important for these truncation selection experiments, any kind of artificial selection, directional selection studies, or attempts to breed more efficient crops or animals that are better for agricultural purposes, you have to establish that there's heritability associated with the trait of interest. All right, I just want to leave with a couple of thoughts about heritability and how it relates to fitness. So remember, fitness is a measure of how good you are at passing on genes to the future generations. And there are traits, which we call adaptations, that are linked to greater fitness. So it turns out that traits that are closely tied to fitness, really adaptive traits, sometimes have really low heritability. Doesn't that sound paradoxical? Because I just mentioned that your ability to have high fitness is related to the genetic basis of you being able to produce adaptations. So a trait that's really closely linked to fitness, how can it have low heritability then? Does that mean it doesn't have a genetic basis? No, it really does have a genetic basis, but it's potentially just been subjected to really intense directional selection for a long period of time. So if you are constantly facing the same environmental challenge and you're forming an adaptation that leads to greater and greater fitness relative to that consistent selection pressure, you're going to be weeding out all of the additive genetic variation. You're going to be making it where the genetics associated with the production of the adaptive trait, they all have the same alleles. In that case, if there isn't any genetic variation left, VA is 0, then heritability is 0, right? If you put a 0 here. So that means whatever phenotypic variation you still have in the population, well, it's due to environmental variation. But it doesn't mean that there isn't a genetic component associated with that trait. It just means that there isn't any remaining variation. There aren't any different alleles that will allow selection to do anything more with the trait. Selection has done so much with it, it's got us to the point where we have low heritability. But again, selection then is going to basically be stopped. It can't change that trait in the future until we get mutation for new genetic variation, new alleles associated with that trait, or perhaps gene flow of individuals that are bringing new alleles associated with those QTLs into the population. All right, so hopefully that kind of makes sense. Well, I'm going to complicate things. If you actually start studying fitness-related traits in nature, you see that sometimes, yes, they have low heritability. Other times, there's actually some heritability still there. So now that should sound confusing to you. Like, well, which one is it? If selection is really powerful and getting rid of all but the best alleles, they should have low heritability. But remember, there are a couple of ways in which you can still maintain, through natural selection, variation in phenotypes in the population. So here are a couple of reasons. Perhaps the best phenotype changes through time. So my previous example was based on the assumption that there was very consistent generation after generation selection pressure, that was the same genetic selection pressure. So big bills are always better. But perhaps the environment shifts so that some years big bills are good, some years smaller bills are good. That's going to maintain additive genetic variance associated with bill size in that trait. We've already gave you an example of that with regard to the medium ground finch, the Darwin finch. A second potential reason why you may maintain some additive genetic variation in a polygenic trait is mutations. Now remember, mutations are pretty rare on a per gene locus basis. But again, we're dealing with polygenic traits here, that the phenotype is associated with many, many gene loci. And if you look at the cumulative effect of mutations across all those loci, there may be enough that maintains some genetic variation in that population. So that's a basic introduction and summary of quantitative genetics, the field of quantitative genetics and the study of polygenic traits. We're going to shift gears a little bit in the next video and go back to talking about adaptations, but we're going to specifically look at different ways scientifically we can form hypotheses, adaptive hypotheses, and then test them. Because if you're going to be a scientist, and evolution is a scientific discipline, you have to form falsifiable hypotheses and you have to find ways of testing those hypotheses.